Okay, so we get the formation of this precipitate, and if we try to quantify how much soluble iron would really be present based on its solubility, so it's focusing on this particular species. Okay, we're going to focus on its solubility product, and it turns out that the KSP, the solubility product, is 10 to the negative 38 molar. And if you were to quantify the amount of soluble iron that would correspond to, okay, at a pH of 7, what that is essentially equal to is a concentration of iron that is 10 to the negative 17 molarity. And that's an extremely low amount of soluble iron. So that essentially would be iron that's bio-unavailable. So the question is, why oxidize iron 2 plus when it is far more soluble than iron 3 plus at pH 7.4? And this is a question in terms of its release into the bloodstream. Well, it turns out that iron 2 plus can generate reactive oxygen species. It's very redox active, and it can do so via Fenton chemistry. So if uncontrolled, this species can cause harm to the body. It can lead to possibly irreversible cellular damage due to a loss of the Ross homeostasis. And so what you can have are cells undergoing oxidative stress. And so in order for you to overcome the high redox activity of iron 2 plus, well, essentially that is the purpose of converting the iron 2 to iron 3. But then you have to then regulate the actual transport of that metal. Because if you don't, there are a few things that are possible. One, as I mentioned already, that iron can precipitate as iron hydroxide. And so then once you do precipitate, that metal is no longer functional, is no longer available. Another thing that can happen, it can very easily be reduced and form iron 2 plus. Then that would lead to an uncontrolled regulation of that metal, generating Ross species, okay, even reactive nitrogen species, which can be, if again uncontrolled, quite harmful to the body. But as it turns out, our body has a molecular mechanism in place to be able to control what happens to that iron that's released. And so upon release, this protein, serum transferrin, will capture that metal. There is some speculation that, there we go. There's some speculation of small molecules playing a role in terms of the iron that's present in the blood. But the thing is that in a healthy body, typically virtually 100% of iron will be serum transferrin bound. If you do have an excess of iron, okay, then small molecules can play a role in terms of the speciation of that metal. But then that could lead to a disease condition that is associated with an iron overload. So the role that small molecules play, at least in the human body, in terms of the extracellular environment, so in the bloodstream, is one in terms of what happens when your iron goes in excess, okay, and even saturates serum transferrin. So typically, the pathway that for a healthy body is this, where the iron 3 that's formed and released into our blood will be captured by serum transferrin, and serum transferrin will be the predominant transporter of that metal to our cells and helps to facilitate its biodistribution. So let's talk a little bit about this protein, as it is part of a family of proteins known as the transferrin family. And so the members of this family come in the monolobal and a bilobal form. All right, so what that means is that it is a single unit, okay, but it will compartmentalize into either into these two forms. And so there is a version of the protein, I would say in terms of evolution, a more primitive form of it that is a single lobe, but the major species or form of the transferrin today are bilobal. So a single lobe is approximately 40 kilodalton. The bilobal form is approximately 80 kilodalton. And here I'm focusing on the metal binding site that's located within each of the lobes of this protein. And so here's a zoom in of the metal binding site. And so there are a few things I want to focus on about this metal binding site. Here we have the presence of 4 amino acids that help to coordinate the metal. You have two tyrosines, a histidine, and a aspartate. And then you have a small molecule, which is carbonate. And that small molecule is very important in our bloodstream as it helps to facilitate buffering in our bloodstream. So we're aware of that capacity of the carbonate. But in the context of metal binding to this protein, it serves as a synergistic anion. And by synergistic, I mean it helps to stabilize the coordination of this metal to this protein. So via this coordination of iron, and that's true for both metal binding sites, okay, so they're, in terms of the coordination of the metal, they're identical. The protein plays a number of functions. It helps to transport iron to our cells, but also to regulate the homeostasis of this metal. And it can play a role of bacterial stasis. And so bacteria thrive on iron. So I've mentioned that in our previous lecture. Bacteria, as I discussed, they can even form small molecules known as siderophores to help to mobilize iron and to be able to transport it into their cells. So what human serum transferrin does, it serves as a strong chelator of iron. So basically, it scavenges the metal as best as can away from, let's say, pathogenic bacteria. And so that also plays a form of nutritional immunity. In terms of its iron binding capacity, well, it can bind iron at very high affinity to both of these sites. And so they are comparable but not equal. All right, so iron binding to the C lobe has a affinity, a log K of 22.2. Iron binding to the N lobe has a log K of 21.3. So I did mention that both binding sites are identical in terms of the coordination of the amino acids and the carbonate. But it's the secondary sphere of coordination. So all those other groups that are surrounding this metal binding site that really helps to influence this affinity. All right, so this characterizes a few features about the iron binding. And so this relates to things that we already discussed about metal coordination and some of the really important properties, specifically the spectroscopic properties. So here is a picture of iron-bound transferrin, and it has this very distinctive salmon-like color. And so the question is, what is the source of this color? Well, it turns out that that color that we can perceive at, let's say, micromolar concentration is due to a ligand to metal charge transfer. And so that particular absorbance is because of the coordination of the tyrosine amino acids to the iron 3. All right, so you have that exchange of electron density from the tyrosines to the iron upon excitation. And so you have this very distinctive visible spectrum. So the apoprotein does not absorb in this region at all. But then when iron binds, you have this very distinct absorbance, and that is the ligand to metal charge transfer. So if we were to measure the extinction coefficient of this coordination, well, depending on how you measure it, so with respect to the protein, this extinction coefficient would be 5,000 per molar per centimeter, and that's characteristic of a ligand to metal charge transfer or a charge transfer in general. If we look at it from the context of iron, remember that this protein binds two equivalents of iron. This extinction coefficient is 2,500 per molar per centimeter. All right, so that's just some of the spectroscopic properties of the metal bound to the protein. All right, so in the 90s, there was efforts to try to understand the general metal binding capacity of this protein. So in terms of the use of this protein, it is the main iron transporter, but it does have the capacity to bind other metals. In particular, it is a hard Lewis acid metal binder. And so what we see here is a plot that was generated correlating the affinity of different metals to the first binding site of the protein. Okay, so depending on the metal, that first binding site could either be the C lobe or the N lobe. All right, so I'm not going to be specific as to which of the two lobes. But that correlation is made based on the affinity of these metals for the hydroxide anion. So there was this linear correlation that demonstrates that the metals that have the highest affinity to this protein are those metals that are hard Lewis acids. All right, so let's talk a little bit about the structure of the protein as it relates to iron binding. So as it turns out, iron coordination to both metal binding sites results in a dramatic conformational change of the protein. And so we can perceive this looking at the apotransferrin structure and the holotransferrin structure, a big angle change with respect to the subdomains of each of these lobes when iron is coordinated. So we have approximately a 50-degree change in those subdomains because of the iron binding at the C lobe, and approximately a 60-degree change because of iron binding to the N lobe. So iron binding does something really dramatic in terms of the structure of that protein. And that structural change actually serves to stabilize this protein. And as we'll see, it also serves to stimulate the recognition of this complex by the transferrin receptor. All right, so let's focus in a little bit on the metal binding residues. So here I'm highlighting the metal binding residues of the N lobe, so you can get a perspective of the positioning of these amino acids. And so you can imagine dropping in iron into the center, okay, how that results in a sort of a Pac-Man-like closing of the lobes. Because now you have to bring those amino acids that are located in different components of the subdomains of this lobe in order to facilitate coordination. The same thing is true for the C lobe. The location of these amino acids with respect to the subdomains of this the C lobe. All right, but there are additional features, and as I mentioned to you, which can account for the difference in the metal affinity to both sites. And so here I'm highlighting the important features surrounding the primary coordination sphere. So primary coordination sphere means those atoms that are directly bound to the metal, and then highlighting the secondary interactions that can influence this. For instance, the carbonate anion, okay, so here we have the carbonate anion, and over here we have it in the other lobe. Well, it has its own pocket that enables its stabilization. So you have an arginine residue that serves as a binding site for that carbonate and helps to stabilize that carbonate. In addition, you have in the N lobe these two really important lysines that are close to the metal binding site that also help to stabilize the close conformation. In the C lobe, we have these three amino acids, this lysine, this arginine, this aspartate, that also helps to stabilize the close conformation of that metal. I'm going to talk about that very briefly. All right, so focusing on the N lobe, as I mentioned to you, there are two really important lysines, and they are basically facilitate what is known as a dilysine trigger, and that is a trigger that helps to facilitate opening of the lobe. All right, so let's talk a little bit about what happens due to iron coordination. So the iron coordination to the metal binding site results in a lowering of the pK of both lysines. So keep in mind, the iron is not coordinated to either one of the lysines. These lysines are simply in the vicinity of the coordination site. But nonetheless, the structural changes that result have an impact on the pKa of these lysines. So normally, the pKa of lysine is 10.53. So at a pH of 7.4, you would have a positively charged amino group for the lysine. All right, so let me just clean this up so you guys can see this a little bit better. All right, so just keep in mind the relationship between pK and pH, right? So if the pH is lower than the pK, then that particular residue will be protonated. If the pH is greater than the pK, then it will be deprotonated. And just to give us a perspective of what we should expect to see. However, when iron is bound to the protein and you have this closed conformation, what you have is one of the lysines deprotonated. And so now what happens is with that remaining proton facilitates a hydrogen bond between the two lysines, and that helps to maintain this closed conformation. Okay, so this hydrogen bonding interaction is stable even at a pH of 5.5. And that's pretty dramatic because at that pH, both of these lysines should be protonated, but they're not. Okay, and so another thing that helps to facilitate this is this hydrophobic box that is created by the metal binding residues. So the the 2-tyrosines, and the histidine. They help to stabilize that hydrogen bond between the 2 lysines. So just to remind ourselves, just a few biochemical notes here. So we're talking about hydrogen bonds, and they can come in the form of a linear interaction between a donor acceptor or a bent interaction, just as a representation of this visually. Typically, the linear ones will be considered the stronger hydrogen bonds. And so when we're thinking about the length of these bonds as they correlate to bond strengths, okay, you get a perspective of the lengths of 2.2 to 2.5 angstroms that are relatively strong, okay, and they are approximating a covalent interaction, and then the longer this length is, the weaker that interaction is going to be. All right, so I would say that bond lengths can be a little bit misleading because it would be hard to know unless you really look at the structure what modality this hydrogen bond is, whether it's linear or bent or something approximating one or the other. Okay, so what does a dilysine trigger actually mean? What that means is that any sudden influx in protons or acidification would disrupt this interaction and lead to the trigger of a conformational change. And so we're talking about the protein going from a closed conformation then to an open conformation. And so I would say this used to be the thinking about iron binding to the protein. But more and more, we're seeing structures that help us to really expand our understanding of metal binding to this protein and what are those factors that may result in metal transport or release. And so here is an example of iron bound in an open conformation. Okay, so this is iron bound in an open conformation. And so you can kind of think of this as a snapshot of the iron in an open conformation, possibly leading to its release. But what's facilitating this is the presence of an anion that is bound to the metal and is preventing coordination of some of the amino acids. All right, so here we get a perspective of how anions can help facilitate this open conformation and possibly the release of the metal. And so in the C lobe, we have something that's very similar as what we observe in the N lobe, that dilysine trigger that can result in the opening of the protein. Well, on the C lobe, there's this pH-sensitive triad, and so those are the amino acids that I referred to you previously. And so what happens is you have a lysine, an arginine, and an aspartate group that are very near the metal binding site of the C lobe, okay? And they have this hydrogen bond network or electrostatic network that allows them to have a strong interaction with one another, that also helps to keep this respective lobe closed. So acidification of this site can also lead to a disruption of this strong interaction and thus an opening of the C lobe. But something that I want to really highlight is differences in the stability of iron bound either to the N and to the C or the N lobe can have an impact in terms of the overall protein structure. All right, so as I've mentioned to you, iron binding in what we will consider the traditional format results in this really compact structure, and so the iron-free form is not as compact. Right, so what we can think about is to try to examine how metal binding can really help stabilize. And so there are methods that can help us to measure this, to examine the stability of the protein in the presence and the absence of the metal. So we're essentially looking at a way of gauging a folded protein or an unfolded protein, not necessarily denatured. So here I'm presenting to you some thermal data. This is differential scanning calorimetry, which can be used to characterize the stability of a protein. And so what I'm showing you here is data that corresponds to just the metal-free protein, metal-free APOM. That's what the terminology that is used. And so you have two distinctive signals. As you increase the temperature of the protein, you get the first signal that is attributed to an unfolding process occurring at the C lobe, and then you get another signal so the first signal was at about, I would say, let's say 58 degrees, and the second one is at about 68 degrees. Well, that second one is due to an unfolding that's occurring at the N lobe. All right, so we have these what are called melting temperatures. But let's see what happens in the presence of iron. And so here I show you titrations of the irons at different equivalents. So here we have 1 equivalent and 2 equivalents. And we see this dramatic change in the profile of the protein. So here again are the 2 thermal temperatures, the melting temperatures of the protein. And then what happens when we add 1 equivalent, we see the loss of this peak and the raise of a melting temperature at about 90 degrees. And then upon the second equivalent, we only see 1 signal. All right. So that is a correlation between what's happening to the structure of the protein when the metal binds and what's happening to the stability of the protein. All right. So the first equivalent of iron binding, okay, is resulting in a dramatic change in the thermal temperature of the C lobe. And this is a huge change, about 30 degrees, very dramatic. Then you have an associated cooperative change in the N lobe of about 5 degrees. When the second iron binds, okay, you also have the N lobe undergoing a melting temperature of about 87 degrees. And this is also a dramatic change. It's about 20 degrees difference. So iron binding to this protein is extremely important in terms of the structural change, but also the stability of the protein. All right, so take-home message here. All right, so if we want to oversimplify this, right, this idea of protein structure and arrangement in terms of its correlation with stability. So at least in the context of transferrin, that more compact structure leads to a more stable protein. And so here we get a perspective of a single lobe, so here the N lobe, for the metal-free form, and then the metal bound to one lobe, and in an open conformation, and then metal bound to both lobes, and in this case, this is the N lobe in a closed conformation. So here we're looking at the N lobe in the open conformation and then the closed conformation. Okay, so here we expect an increase in stabilization of the protein. So students from last year's course, well, they did a study about this correlation between metal binding and the conformational change. And so one of the highlights of the work was identification of what can be classified as the canonical binding. So this is a term, well-known terminology. And they also defined what constitutes a non-canonical binding. So essentially, the form of iron or other metals that can coordinate to this protein that is unlike the typical iron-bound form to this particular site. Right, so I have already shared you guys that reference. But in terms of what role does the serum transferrin play with regards to iron? So we know that the iron improves the stability of the protein, but what does the protein do for the iron? Well, the protein actually enhances the solubility of that iron. As I already mentioned to you, the iron solubility would be extremely low if you were just to put it in the context of water, blood, pH 7.4. But here, where we can observe iron, in terms of its concentration in blood, is a concentration of 22 micromolar. And that is attributed to the interaction or the binding of the serum transferrin protein. Okay, and so we also get a significant amount of iron that gets delivered into our cells. So we can even reach, I would say, low, even probably approximately mid micromolar amounts of iron. All right, so this gives us a perspective about the biodistribution of that metal. So the intake of iron, okay, so I already saw the video of iron coming in through cereal or other ways in which iron can enter, then iron being bound by transferrin, and then distribution of that transferrin. Some amount is going to the hemoglobin, which constitutes our red blood cells. Within cells, there is the ferritin protein, which will store the metal. There are other processes that will utilize that metal. What we observe typically is a balance between how much iron is absorbed and how much iron is basically released from our bodies. I will say that there is no true physiological excretion method for the metal. The typical manners in which iron is released from our body is basically our skin shedding. So if you scratch your skin, you're going to lose some iron in that process. But we don't actually have inside our bodies a sort of a molecular mechanism that can control the release of the iron from our bodies, right? So that is why it's very important to control the amount of intake. Because if you intake too much iron, well, that iron is simply going to accumulate in your body and can cause an iron overload and toxicity associated with it. All right, so transferrin bound iron is the dominant iron form in the blood, but some non-transferrin bound iron can form, but it has to be well regulated, because that can lead to, again, as I mentioned previously, oxidative stress as a cause of a Ross species. All right, let's talk a little bit about this idea of how exactly does serum transferrin enable iron uptake into our cells. And so this is a receptor-mediated endocytosis pathway. So we talked a bit about what is endocytosis in our last lecture. And this is a process that involves redox, and there are a number of key players. So there is the apoprotein, there is the iron-bound protein or the hollow protein, there are the transferrin receptors. I'm going to focus on the transferrin receptor 1. There is clathrin, we briefly talked about it last time. Iron chelators, so these are biological chelators, a protein called STEAP3, and even the divalent metal transporter. All right, so these are key players in this process. All right, so the steps that are involved. All right, so you have a pH-dependent process that modulates apo and hollow transferrin affinity to the transferrin receptor. All right, so that's something really important to think about. Then you have the formation of an endosome, and then how that endosome gets delivered into the cell. Then you have an ATP-dependent pump that's going to lower the endosome's pH from a pH 7.4 to a pH 5.5. Then you have iron release from the protein, and finally iron release from the endosome into the cytoplasm. All right, we're going to look at this in a moment. We'll look at this in terms of the interaction with the receptor. So here is a visual of the transferrin receptor. So this is a transmembrane protein, so it has its different compartments. The ectodomain, the part that is the transmembrane component, and the cytoplasmic region. So this receptor is homodimeric, and it's found at the cell membrane, and it binds at maximum 2 molecules of iron transferrin. At a pH of 7.4, this receptor has a preference for binding iron-bound transferrin. So it has a relatively high affinity for this protein. Okay, it does not have a high affinity for the apoprotein. All right, so that's something really important to note. At 7.4, the receptor requires interaction with iron-bound, and that interaction results in a signal to then begin the process of endocytosis. But at a low pH, so a pH of 5.5, that affinity actually changes. The receptor no longer has a high affinity for the iron-bound protein. Rather, it has an affinity for the apoprotein. So it has a preference for basically releasing the iron. Okay, so here are a few things that we're going to focus on. So there are different proposed mechanisms. I would say that even today, as well studied as this protein is, there isn't a true agreement about how the iron is released into our cells. So this is one proposed model, a chelator-dependent model, where you have an acidification event, and then you have a chelator that takes advantage of that acidification to bind the iron, and then releases the iron from transferrin, and then that iron gets reduced by the this reductase STEAP3. All right, let's look at a visual of this. Okay, so here is a cartoon representation. You have your iron transferrin gets recognized by the receptor. Okay, then you have this receptor becomes clathrin coated. Okay, remember I discussed clathrin. Clathrin basically helps to protect the endosome. And the reason for this is because you don't want that endosome to get proteolyzed. You want that endosome to be able to return to the membrane so that the receptor can be recycled in order to capture more iron. All right, so once the endosome is formed, then you have this ATP pump that's going to lower the pH of the endosome. So now you have a pH of about 5.6, 5.5. That acidification step appears to be coupled with the binding of the iron or the release of the iron from the protein. So there could be chelators that become present and start to bind the metal. So they maybe it's forming some metal complex. So maybe you have some iron. Sorry, I was trying to draw an iron metal complex that forms. Okay, but whatever the species is, that iron as iron 3 plus will then get reduced to iron 2 plus. So you have this iron that now gets reduced into its iron 2 plus, and then you have the divalent metal transporter here, which will then shuttle the transport of the iron into the cytoplasm, and now the iron is able to be utilized. Okay, so that's a chelator driven pathway. But there's another pathway, which is one that results in a lowered metal affinity to the protein. A very similar pathway, but this has to do with the oxidation state of the metal. And so here I'm presenting you, this is a figure that you can find in a typical textbook that helps us to understand what is known as the biological redox range. So we're talking roughly around negative 500 millivoltage to positive 800 millivoltage. All right, and you have those outliers that can go even further. All right, so this is a range that can facilitate whether something might be reduced or oxidized. Well, according to this particular mechanism, what happens is the redox potential of iron-bound transferrin is modulated by the receptor. Okay, what do I mean by that? Well, when you measure the reduction potential of the iron, so reduction of the iron from iron 3 to iron 2, okay, so this is a 1-electron process. Okay, and then will be 2 electrons, obviously, because you have 2 metals here. Well, that is a potential that is quite negative. But it turns out that in the presence of the receptor, this potential is more positive, so it's still negative, but it's more positive, and is believed to be within the physiological range. And that STEAP3 protein appears to be able to facilitate that, that process of reducing the metal when it's still bound to the protein. But why is that important? Well, that's important because that change in oxidation state is enough to dramatically alter the affinity of the protein for the metal. As you can see, there are many orders of magnitude difference between how strongly that protein binds iron 3 and how strongly it binds iron 2. And so this lower affinity is enough to then remove iron from the protein as iron 2, and then that iron 2 is transported out via the divalent metal transporter. All right, so this is a redox-oriented process. Right, so here's just the visual to help us to see this. Okay, this idea of a reduction of the metal when bound to the protein already, and that is enough to then release iron as iron 2 plus, and then out into the cytosol. All right, so that is a proposed mechanism. Again, there is no true agreement between one or the other. I would say that there's probably a higher agreement for the first pathway than there is for the second pathway, just given the nature of where this interaction between the iron transferrin transferrin receptor and the STEAP3 protein might be possible. Okay, so I'm going to skip over this. I will actually touch upon this in our next lecture. So these are things that were highlighted by students from the last year's course. The main thing I want to focus on before Manoj will take over is this idea of what happens to that iron once it is actually in our cytoplasm, in our cytosol. All right, so this process of endocytosis is really quick. So for immature red blood cells, it happens within 2 minutes. And so what you get is the formation of what is known as a labile ion pool. And labile means very reactive, and is actually temporary. So some of that pool will get stored in the protein ferritin, and some of that pool will get used incorporated into really important proteins and motifs. Okay, and so we're going to look at this utilization of iron. And so some of that iron gets trafficked into the mitochondrion and gets incorporated into the protoporphyrin 9 to form heme. And so I'm going to stop there because it's just the introduction to this idea of the incorporation of that iron into a motif that's recycled, actually very significantly, in many heme-based proteins. All right, so I'm going to stop my projection here. Okay, and I'm going to quickly introduce Manoj. Manoj, in the meantime, can you load up your slides?
Let me see.
Okay, so while Manoj is doing that, all right, so it is my honor to introduce you to Manoj Saxena. So Manoj is originally from India. He obtained his bachelor's in chemistry in Dove College, and then an MS in biotechnology, and also an MS in biochemistry, microbiology, molecular biology at Pennsylvania State University. He obtained his PhD here at the University of Puerto Rico in environmental sciences under the mentorship of Dr. Kai Gribenow. And for a brief amount of time, he served as a postdoc in my laboratory. Since then, Manoj has done postdoctoral work at Matthew Coleman's laboratory in the Department of Radiation Oncology at the University of California, Davis. And now he is currently doing a postdoc at Birkbeck College, University of London, in the laboratory of Philip Robinson. So Manoj has extensive expertise in biochemistry. He did work in cytochrome C, which will serve as the basis of the things that we're going to focus on in the course. And he has extensive skill sets in X-ray crystal structural characterization. So he was actually a really important person in my laboratory in solving a crystal structure that really helps us to better understand the metal binding capacity of transferrin. All right, so with that introduction then, Manoj, you're the star of the show.
Thank you for the kind introduction, and I will start my presentation now. Good afternoon, everybody. I hope everybody can hear me properly. Can somebody confirm it?
Yeah, we can hear you.
Okay, great. So thanks for the invitation to give this talk, and it's really a pleasure to come back to my university where I did my PhD, and although even virtually because of the COVID conditions, as you all know. And so today I will talk about cytochrome C, for which Dr. Tinoco has given a very good introduction in terms of iron binding proteins and how iron is incorporated in the heme. And also today I will talk a little bit more about the work that I have done as a PhD student in Dr. Kai Gribenow's lab in UPR, where I have utilized a specific property of cytochrome C to use it as an anti-cancer drug. So during this talk, I will give you a brief overview of cytochrome C, its properties, in terms of how study of cytochrome C itself has evolved and how the specific study of the sequence of cytochrome C comparison among different organisms gave rise to evolution of a new branch of studies called evolutionary phylogenetics. And also I will give you a brief idea about what are the different roles of this cytochrome C in electron transport chain, as well as a trigger for apoptosis or cell death. And so this protein is a very small protein compared to many proteins which are even megadaltons in size, only 12 kilodalton. So we can say that this is a robust protein with a multifaceted personality. And here you see in this diagram a space filling model of this protein, and also the heme pocket is shown here, where this heme is the prosthetic group here and coordinated by iron and different protein residues, which I will talk later about. So as I said, this is a small protein of only about 12 kilodalton in humans and about 105 amino acids. And here is a cartoon representation of this protein. So when the study of this protein was just beginning in early 1950s or late 1960s, during that period, there was a burst of different studies that started about this protein because of work of Emmanuel Margolius. He actually sequenced many of these proteins, and starting with about 20 proteins, he compare all those amino acid sequences, and what he found that if you compare the sequence of this protein among closely related, evolutionarily related organism, you will find a complete match. But as you can see in this other panel here, where I have compared the human sequence with horse and budding yeast and rhizopus, all the cytochrome C, if you compare, you will start to see that there are differences in the amino acid sequence. So all this study uh, gave rise to a new science of molecular phylogeny, in which you can compare the sequence of proteins and determine how different organisms are related to each other in evolutionary terms. So in this slide, you are looking at a diagram which is showing you a evolutionary relation between different organisms uh, ranging from human to Neurospora, just based on the sequence of the gene that is responsible for cytochrome C. So this was one aspect of early studies of the sequence of this cytochrome C, which gave rise to a totally new branch of study called molecular phylogeny. Now, moving more closely into the protein structure, as was referred by Dr. Tinoco in his talk, that there is a prosthetic group called heme-C, which is part of this protein. And here, depending on how this prosthetic group is attached, there are different classes of heme. And in case of cytochrome C, this is called heme-C because of the way it's attached. So this line denotes the protein sequence, and this is iron, which is in the middle, and they're not shown in this figure, but there are two axial ligands of also present in the protein. So this is the way the heme-C is attached to the protein. And also among the heme-C, there are also different classes, which heme-C is further classified into 4 different classes based on how its protein is attached to the protein and how many of those heme-C groups are present. Because there are certain proteins in which there are multiple heme-C groups, which can be seen on this panel here in the classes of heme-C. And many of the properties of this protein cytochrome C are attributed to this heme-C itself, and specifically the iron. The ability of this iron to change from one oxidation state to another, namely ferric to ferrous, or this is a reversible reaction, so they can change both ways. And because of this property, one of the important aspects of this protein is that it's part of the inner mitochondrial membrane, which you are looking at the figure now here, where there is a chain of different complexes in this inner mitochondrial membrane, which constitute a very important electron transport chain, which is the basis of all the energy formation inside the cell. Because when the electrons are transported in this chain from one complex to another, a proton gradient is formed, and the cytochrome C is part of this chain, where it actually transports the electron from a complex 3 to complex 4, and thereby constitute a very important part of this electron transport chain. And as you all know, probably from your biology classes, that ATP is the currency of the cell, and this electron transport chain is crucial for producing all the ATPs, because that's the end product here, where this ATP is formed by the complex 5 of this electron transport chain. So although this is a very important function of cytochrome C, in today's talk I will focus on a different function of cytochrome C, which is not related to this, which is its ability to trigger apoptosis. So cytochrome C not only plays an important role in electron transport chain, but it also has another role which was relatively more recently discovered, that if this cytochrome C, which normally reside in this inner mitochondrial membrane loosely associated, if somehow due to cellular stress it reaches the cytoplasm, it can trigger a reaction, a chain of reactions called a caspase cascade, whereby different caspases, which are actually proteases, they get activated, and in turn what they do is they are like molecular scissors. They go on and cut different proteins that are essential for cell and induce the cell death. So this ability of cytochrome C to induce apoptosis in cell was noticed by many, and some people actually did experiments where they actually injected cytochrome C into the cell, because normally the cytochrome C in vitro in conditions you cannot just inject into the cell without actually injecting. The uptake is not possible because it's impermeable. So this gives an idea that it can be a potential anti-cancer drug, because cancer is essentially an uncontrolled growth of cells. So if you can kill specifically the cells by introducing from outside cytochrome C, you can kill the cancer cells. So we will see this how I developed this idea more into my PhD project in next few slides. So as I said that cytochrome C can induce cell death, and this action happens by activation of certain enzymes called caspases. Among these, caspase 3 is one of those enzymes, and on this slide what you're looking at is that even if you take the cytochrome C from different sources, sequences of which are compared here, human, horse, or rhizopus, which is a form of fungus, you will see that if you compare the activity which you see in this graph, that you will see there is like a difference in activity of this caspase activation, but there is activity, and this activity relates to how well these two different cytochromes matches in their sequence. So horse and human have very comparable activity. So this idea of evolutionary conservation also is conserved in terms of its activity in for activating the caspases for inducing cell death. So based on all this information, that if you introduce cytochrome C from outside, you can induce cell death, and you can use this cytochrome C to activate the caspases. I build upon this idea, and combining with the fact that this protein on itself is not permeable, I needed some other carrier protein to transport it inside. So this just from previous few slides that were presented by Dr. Tinoco, you learned about this transferrin as a carrier protein which carries iron. So I will not go into detail of how it carries iron, but on this slide what I'm showing, and the emphasis is here that if you compare the normal cells or the normal tissue to the cancer tissue, you will see that there is a preponderance of the receptors of transferrin, which is shown here by dark staining in cancer tissue compared to the normal tissue. So because of this, you can specifically target the cancer cells if you can somehow combine transferrin with your toxic or apoptotic inducing protein like cytochrome C. And to further visualize or to further see if this idea is feasible, I actually checked A54 cell line, which is a lung cancer cell line, to see if I can visualize the transferrin inside the cell. So here, in this slide, what you are looking at is that how I label this transferrin with a dye which gives its green color. The green color is due to FITC. So using a technique called confocal microscopy, I looked at these cells, which is A549 lung cancer cells, and by looking at different depth of the cells, which is B and C denotes different depths, I could see that actually when you incubate these cells with transferrin, it actually goes inside. So this was like to further validate that in my hand, this A54 cell line, when incubated with labeled transferrin, can actually uptake transferrin. And because of all these properties, transferrin can act as a perfect delivery vehicle for this apoptotic protein cytochrome C. And also it has been already proven by different groups as well as by our studies that not only it takes its canonical load, which is iron, it can take other small metals, as well as it can deliver DNA and small molecules. So encouraged by these previous studies and by my experiment of uptake of this titanium of this transferrin in A549 cells, I further designed a system by which these two proteins, one cytochrome C and this transferrin, were attached together for uptake into cancer cells. So this objective of combining these two proteins was designed so that I can target specifically the cancer cells to induce cell death. By combining the properties of these two proteins, transferrin will do the targeting and cytochrome C will induce the cell death. So this is the overall picture which summarizes the design concept in which these two proteins were combined in a chemically cross-linked way. And when these two were combined, they were attached in such a way that they will go inside and then separate because of the reducing environment inside the cell, and then induce apoptosis in the cancer cell. So this is something which I will tell you in more detail in further slides. So this slides gives you the three basic steps in which these two proteins were combined. So as I was speaking before, that I use a chemical cross-linker which combine these two proteins in such a way that these two proteins become attached by a disulfide bond. And as you know, that in reducing condition, these disulfide bond will convert into sulfhydryl, and these two proteins will become apart. And that was the aim, so that this protein cytochrome C alone goes in when it's inside the cell, will do its action of inducing apoptosis. So once this protein were combined together using this chemical cross-linking method, what I did was I purified this complex using a technique of chromatography called gel filtration chromatography. As many of you might know, that in gel filtration chromatography, you get the bigger molecules first and the smaller ones later. So this was done to separate the formed conjugate from the unfinished reaction, because any reaction is not 100%. So there was some unfinished or unconjugated product, which you can see on the gel here, and the finished product, which contained both transferrin and conjugate attached together, was diluted. And this was further characterized to show that these two proteins are attached, and to also show how many proteins are attached together. Because as you have seen in previous slides, and also Dr. Tinoco mentioned, the size of transferrin is about 80 kilodalton, whereas the cytochrome C is smaller protein. So there is a possibility and that multiple cytochrome C will bind to transferrin. So to see what is the stoichiometry of this complex found by conjugation, I used a technique called mass spectroscopy, MALDI mass spectroscopy. And here you are looking at different spectra where peaks are labeled. Different peaks are labeled according to the number of different cytochrome C bound to transferrin. So because there are different stoichiometry, and the complex found was a mixture. But one important aspect that we designed into this complex of cytochrome C and transferrin was that once inside the cell, this conjugate should be released. So we designed it in such a way that because of this disulfide bond, in acidic condition or in reducing condition, which are the normal conditions inside the cell, that this disulfide bond will get broken into sulfidyl bonds, and then the two proteins can get separated and perform their action. So here what you're looking at is the result from a dynamic light scattering experiment in which the conjugate or cytochrome C alone is compared with the conjugate which is incubated in conditions which are inside cell, because inside cell is a reducing condition and glutathione is a chemical which provides this reducing condition. Also, when I incubated this conjugate in acidic condition, what you can see that the two peaks are separated, which indicate that these two proteins were separated on incubating in acidic condition as well in glutathione. And this result was further validated by using gel filtration chromatography. As I explained before, the principle of this gel filtration chromatography, so that you can see that before incubation, this conjugate was mainly one peak, and after the incubation in the reducing condition, I can see two different peaks denoting the two constituents of this conjugate. So by using different techniques, I was able to validate that this conjugate system, if enters the cell, this will break into two independent components and can perform its function. Next, after doing all these in vitro experiments, our focus was to show that this conjugate actually works in cells. So these studies were done on cancer cells, specifically the lung cancer cell A549, and what you're looking at is a panel of different cells in which the cells were treated either with a control, that means there is no conjugate, just PBS, which is a buffer, and or the cells were treated with the conjugate, or you have cytochrome C alone treatment. So if you compare them, what you see that cytochrome C was probed by a green antibody. So if you see that in untreated control cell, it's just punctate or very pointed cytochrome C, because this is just the endogenous cytochrome C label of the cells. But when you treat these cells with conjugate, there is an influx of cytochrome C inside, and there is a lot more green color here. And when there is cytochrome C, which is just fit C label, was treated, because cytochrome C on its own is unable to enter the cells, you basically see a very faint or almost nothing here. So also in here you see that to show the localization of cytochrome C in the mitochondria or outside mitochondria, mitochondria is also tracked by a dye called mitotracker red. So if you compare the conjugated cells with untreated cells, you can see that when the cells were treated with conjugate, there is a lot more conjugate inside, and in the merge you can see that there is this yellow spreading all around as compared to this punctate features here. So a summary of this is that we were able to successfully deliver this conjugate into the cells. But when you deliver any of these drugs into the cell, one of the drawback is that these endosomes are formed, and sometimes your drug is trapped in these endosomes. So an experiment was done to show by specifically labeling the endosomes by a dye that is Alexa Fluor 555, which stains the endosome, and also localizing the cytochrome C. So in this particular panel, you see even in the treated cells, you can see the label red and green are separate. That means the conjugate which was able to enter inside was not trapped in the endosome, but was able to escape the endosome. So that means it was available for its action to induce apoptosis. And some of the other experiments were done to show that this cell, where the cells which were treated with this conjugate has a real activation of the caspases, which is a way by which the cell induces cell death. So when the cells were compared, untreated cells or the cells treated with just the cytochrome C or cells treated with just transferrin alone compared to conjugate, we can see that both caspase 9 and caspase 3 were activated by our conjugate. So one other thing that is very important, was very important to show, is that when you compare the cytotoxicity of this conjugate on cells, cancer cells, and compare it with the normal cells, what we should see, what we should what we want, that this conjugate should be toxic to the cancer cell alone and not to the normal cells, which is MRC5 is the normal cells and A549 is a cancer cell line. So by comparison, we want to show that this conjugate is specifically targeting the cancer cells. So here in this panel A, you see that with increasing concentration of this conjugate, we see that cell viability is decreasing. That means more cells killing with more conjugate. And this killing was as good as a commercially available drug, cisplatin. So, but in case of the normal cell, even with the increasing or the highest dose of 50 micromolar, you can see that there is no cell death in case of this cytochrome C transferrin conjugate, because it's as good as the cell alone. And to further confirm that this cytotoxicity is not just specific for one cell line, A549, we also tested HeLa cell and we got the similar results. And to further validate that this cytotoxicity is acting through a mechanism of apoptosis activation, we tested another cell line, which is K562, in which apoptotic pathway is blocked. So if this pathway is blocked and our conjugate is acting through this pathway, we expect to see no cell death. And when we compare, we saw that in K562 cells, we found that almost 100% viability in the cells which were treated with the conjugate, further validating that this conjugate system was acting through apoptotic pathway. So in summary, what I have shown today is that cytochrome C is a small evolutionary important protein and it's well conserved. It can, and its sequence study can be used to link evolutionary relationship between different organisms. And cytochrome C is an important part of electron transport chain, whereby it transports electrons from a complex 3 to complex 4 of electron transport chain, thus playing an important role in producing ATP for the cell. And it can also induce apoptosis in case when there is a stress or cells are damaged, whereby its release in the cytoplasm can trigger apoptosis. And this property was utilized during my PhD project in making a conjugate of cytochrome C with protein called transferrin in such a way that a conjugate was designed that it will be released once inside the cancer cell. And this conjugate was extensively tested in different conditions to show that successful release inside the cells with different techniques, including confocal microscopy. And we were able to show that this conjugate was effective in cancer cells and was specifically killing the cancer cells. So with this, I end my talk, and I thank members of Kai Grivinov Lab and Dr. Kai Grivinov because this work was done during my PhD work under mentorship of Dr. Kai Grivinov. And I'm really thankful to Dr. Arthur Tinoco for all his help during my stay and my work as PhD student there, and also during my postdoc time that I spent there. And thank you for the invitation, and I can take any questions that you might have at this point.
All right, thank you, Manoj. So does anyone have a quick question?
I have a question.
Sure.
Sure.
I think it's a little elemental, but I don't know a lot about cancer research. But is the apoptotic signaling pathways for all cancers the same? They use caspase 3 and 9 for the initiation of the pathway.
That's a good question. Actually, no. The answer is no. Not all cell lines use the same pathways. As I referred in my presentation itself, there is this specific cell line that I use. In this cell line, that's K562, this pathway is actually blocked. So there are different pathways for cell death in different cell lines. But apoptosis is a very common pathway.
Okay, thank you.
Yeah, sure.
Well, thank you so much, Manoj, and I really appreciate this, and it serves as an introduction to what will be the focus of the review article. All right, everyone, I'll see you next time, and take care, Manoj.
Thank you.
All right.
Thank you both. Have a great day.
Yeah, take care.
Bye.